well, the jock sort of stuff didn't work out. So
clearly did.
I mean, look at me now. Yeah, exactly.
He's got a big gut, and sometimes he sits in.
I call it a thesis belly.
A thesis belly.
I can't think of any more human activity than conducting science experiments.
The game I play is a very interesting one. It's imagination in a tight straitjacket.
The beauty of a living thing is not the atoms that go into it, but the way those atoms are put together.
What I always think should be the basis of education is not answers, but questions. We should teach kids how to question.
Today on the podcast, we've got actually a friend of ours, David Weston.
Your friend, not mine.
Yeah.
So I actually sit next to David. He's a Masters of Research student. He's doing his thesis that's due in about 3 months or so.
Yeah, just under 4 months.
Yeah. So for those of you guys who don't know, the Master of Research is a 2-year program. The first year is all coursework, and then the second year, students kind of work on a 9-month thesis, and you're 3 months away then.
Yeah, around about 3 months. That's cool.
Yeah. Are you sweating it yet?
Yeah, that's what I'm trying to say. No, it's 4 months. It's 4 months.
Yeah, man, and it sucks. I'm sitting next to him and I have to smell all that dirty sweat. I keep telling him, put on some deodorant, dude, some antiperspirant, but he doesn't. This is what I mean, like, he's friends with you because you're like one step away. I'm right next to this guy. We actually had David on the podcast before. When we early on in our podcast journey.
Yeah, when we were still trying to find our feet and get good audio and stuff.
And David, we recorded an interview, and then just so happened that we lost the interview. Wink, wink, nudge, nudge, because it was terrible. No, it wasn't terrible. No. I had to reset my computer, and then I lost a bunch of interviews, unfortunately. And David's one was one of those. So we're happy to have David back on the podcast. A little wiser, a little older, and
little hairy
no less. Yeah, a little hairy actually, and less creepy looking than last year. He's much more approachable.
I think about 10 kilos extra as well. So
okay. Well, let's start this podcast, dude.
Yeah. So you're an inorganic chemist, right?
I'm a lot of different things.
Yeah.
I like to say bioinorganic synthetic chemist.
Inorganic synthetic chemist?
Bioinorganic synthetic chemist. And fluorescent chemist, so I don't know. I guess let's just call it an inorganic.
Fluorescent chemist, does that mean you like glow at night?
Yeah, it does.
It does. It does. It means I'm a chemist, but I glow.
I thought all chemists glowed at night. They hang around so much, nasty stuff.
Yeah, fair. If we're not dying of cancer because of our chemicals, I think we're glowing because of our chemicals. So, and then eventually dying of cancer.
It's like Mr. Burns from The Simpsons, right? Do you remember that episode? Everybody thought he was E.T. and shit, but he was actually getting...
Yeah, see, that'll be me. I reckon once all the mercury goes to my head, that'll be me. Yeah.
Okay, well, tell us about why you became a biosynthetic or inorganic, whatever. just like all those fancy labels to make yourself feel better, chemist.
It does. I think the longer the word, obviously, the more important you sound. But basically, the reason why I came, I don't know, I think from an early age, I kind of had a fascination with things. So I used to obsess over things. I think when I was between the ages of about 3 and 4, whatever, apparently I couldn't stop wearing Batman. And then a Batman costume.
Wait, stop.
Yeah, I'm not kidding.
Couldn't stop dressing up as Batman?
Yeah, my parents used to have to try and force off a Batman costume from me just to watch it. And I'd sulk and cry.
Yeah.
And then I think once I moved to Melbourne, because my family's from the UK, moved to Melbourne and I obsessed with the AFL there. I remember I went through a phase where I started obsessing with astronomy. So that's kind of also a leeway into science, and just had weird obsessions as I go.
I thought you were going to say, I'm a biosynthetic astronomer, goddamn astrobiologist, chemist, but
I didn't quite go that far.
Yeah, good.
Yeah, but I don't know, I think at the same time, I had a lot of fascinations, and I couldn't decide, poor at deciding, And yeah, so I ended up going, okay, what can I do? I want to keep my biology or biochemistry at least, and keep my chemistry, and try and even branch into a bit of medical stuff, which is what I'm trying to push towards, doing some of it for my thesis, so that hopefully in my PhD I can focus on that as well and earn a bit more moolah.
That's cool. It's interesting you say that because about like being driven towards science, you think because you're like had obsessions and got like focused in on stuff.
Yes.
So I think it takes a certain amount of like obsessive compulsiveness. No, not literally. Obviously, that's a proper psychiatric disorder. But you know, scientists have this obsessive personality, don't they? You know.
Yeah. I think I think you're kind of born a scientist in a way. You kind of just naturally. I think I was kind of naturally inquisitive and very easily distracted, very bored at small, simple things. So I had to kind of branch off to more exciting things, and I think that's also what's kind of pushed me to come to the end of my masters and now and even start looking for scholarships for my PhDs. I just kind of constantly want to know more, and I think that's a common trait, an obsessive trait as well for other scientists, not just myself. So
yeah, that's cool.
Yeah, it's an obsessive desire to learn more. I think that's an important quality to have as a scientist.
It also ties into that like nerd culture as well. Like, what are nerds? Really, nerds are people that obsess over stuff, aren't they? Right? Like,
well, I was in jock in high school, man. I used to play AFL throughout all of it.
But were you a jock nerd? Did you obsess over?
I used to. I remember, was it being year 10, and it was around about that time where I was hanging around a lot of girls because I fancied one at the time. And she didn't fancy me back, but that's another nerdy thing. That thing's common. But yeah, essentially that happened, and whenever they come to me with their problems, I kind of just hyper-analyse it and give them a direct answer and say, well, have you tried this? Have you tested this? Sort of thing. And they'd always get annoyed at me for disagreeing with them even. And I think that was kind of probably the first sign of saying, hey, you're still analytical. You've got to do a occupation or whatever that's a bit more analytical and a bit more objective, I suppose.
See, that's the move. David's hustling right from the get-go, using the nerd angle to support the jockey behavior, I suppose.
Yeah. Well, the jock sort of stuff didn't work out.
Clearly did.
So look at me now. Yeah, exactly.
He's got a big gut, and sometimes he sits in.
I call it a thesis belly.
A thesis belly.
No, dude, I didn't get any of that, dude. I had a nice family pack.
Yeah, no, it's because you barely did anything though. During the masters. Just scratched by, try to get a publication.
Just, oh my god. Man, this guy's a savage. Alright, no, that's just fire under the, whatever you call it. I'm just going to go and pump out 10 publications.
Oh yeah, of course you are.
So did you get into, did you do a Bachelor of Science straight after school or what?
Yeah, I did. Yeah, I was very immature back then, and probably spent the first half of
then.
Yeah, still probably am a bit, to be honest. But yeah, I jumped straight in from high school, which I guess I just turned 18, so you tend to get a bit rowdy in your first year, but obviously mature enough and you get into it and start figuring your life out. And yeah, so I think that's the trick I find if there's anyone who's listening to this who's coming through from high school is normally your first year at least you need a bit of time to mature, but yeah, I still did it, got through and got a decent GPA, and now look at me. So
yeah. And when you decided to do a Bachelor of Science, were you doing that because you wanted to do scientific research, or did you have another career in mind, or what? Why? I guess that's a big move, right? You're sitting there as a HSC student, and then you pick. What did you do? Bachelor of Science, medical science?
Oh, I did a Bachelor of Science double major in biochemistry and chemistry, but I did a bit of medical back then as well. But now I'm getting more medical.
So that's that's a big decision. What career goal? What was your end goal? Why are you doing this course?
All I really wanted to do was be called doctor. I'll put it out like that.
Join the team, man.
Yeah, exactly. That's why we're all here.
You guys are pathetic.
Yeah, we are.
Stop judging us, dude.
You're doing the same. Come on,
parental
man.
pressures, dude. My mum always wanted a doctor, and so all right, I'm not going to be a real doctor, so I'll just be a fake one. With
be a research doctor. Yeah. But I don't know. For me, I guess when I kind of knew that I went to a Bachelor of Science, probably around about year 11. I remember, year 10, I think I topped my class in science. And so for when I was choosing my electives, I chose biology because I knew I wanted to do biology. And then I actually chose chemistry as the one underneath, and I was encouraged by my deputy principal to choose chemistry and drop what's it called again, hospitality, which I was going to do. And I kind of went, oh yeah, okay, great. So I only really chose chemistry because I was good at it. And then when I got into year 11, I think you start learning some real chemistry, which I don't really think you do before then.
Yeah.
Then I started to realise that this was kind of my calling, I suppose, and knew that I wanted to do science. And yeah, and especially throughout my bachelor's, I kind of realised that chemistry specifically is for me, and obviously I didn't want to drop that biochemistry at the same time. So yeah.
Why chemistry in particular, though? Because sorry, let me just clarify, because I was in a similar boat, right? I started my undergraduate here, and I was kind of tossing up between the majors, a biochem, molecular biology major, and then going the chemistry route, because I love chemistry. There's something about chemistry that really fascinates me, because at the heart of it, and I like biology too, right? But at the heart of it, biology is just more complex kind of chemistry. That's how I see it. So by understanding that at the fundamental level, you're kind of understanding like the real nature of biology by understanding chemistry. Yeah,
well, essentially, I just putting it out there, I kind of see chemistry is what I love, and then biology is my application. And really, just doing hard chemistry, I suppose in the big world, there's not too many opportunities out there. I mean, there's still some. There's not too many, and that's why I like medical and biochemistry, which to me are kind of, well, medicine's kind of glorified biochemistry is what I like to call it. They're kind of the applications for chemistry, I find. But I enjoy both. I definitely enjoy my chemistry more. I found learning it in bachelor's is a bit more analytical. Learning biochemistry and biology was a bit more, here's this word or here's this pathway, now remember it, but not so much remembering or knowing why this chemical changes to this chemical, which is what I find interesting, the mechanisms of how things work. But yeah, the actual sitting down and memorizing things, I don't necessarily have the best memory as it is. So
Well, I agree and disagree with that. I think you're right about like the chemistry thing, like understanding the actual mechanism. Like that's what really excites me about chemistry, and why in my early stages I was tossing up between the two. But I'm actually like you. I have a terrible memory, right? But I think you can do.
Yeah, he's probably got dementia by now.
Yeah. So I kind of disagree. Obviously, I'm a biologist, so I don't quite agree with you have to learn everything by rote. I got by. You can. There's some basic principles, I think.
But if you ask me, some of the best biochemists and medical scientists are all chemists, really. So
because
we actually know what we're doing. So.
Ouch. The gloves are off. The gloves are off.
They are. I mean, the purple ones are on.
Well, it's funny because Alex, like, over here is just like, oh yeah, I'm such a big fan of chemistry. Chemistry is a shit. It's so great. Blah blah blah. And then when we go back into the postgrad room, he just starts talking shit to all the chemists about how great.
And pretending he knows what we're doing.
That's just how I show my love as an Australian, right?
You just roast the ones you love the most.
Well, that probably explains a lot of your behaviour then, Hamid.
Because
I think I probably cop more from anyone in the postgraduate room from you.
I'm just feeling the
this
love
guy's
right
essentially
now.
a bully, and I think that's what your blab coats fellas don't realise.
No, I'm not smacking you around with a bat. It's with a pillow, man. It's a pillow of love. Just with a lot of criticisms that pillow's filled with.
That's fine.
And
sometimes I need it anyway.
It's just to keep you humble, dude.
So maybe I don't know. What do you?
Let's transition into your research. Okay, so you said you're inorganic, biosynthetic,
and fluorescent.
Something fluorochemist.
Yep.
Who does astronomy on the
and
weekends.
Likes AFL.
And likes AFL, but he also does like runs and shit. So
I haven't
used to run if anyone's watching the video, you would know that David clearly doesn't run anymore, because like that
run much,
that gut of his is like it's worse than Homer Simpson's. But he's a handsome young man. He's in a relationship. He's secure and safe. So he doesn't have to make an effort anymore.
Yeah, exactly. To be honest, I think that's half what it is. I'm writing my thesis. I'm in a relationship. I don't need to care.
Oh, dude, I'm putting on weight as well. So.
But let's hear about the ligand.
Yeah, the ligand.
Yeah. Oh, no, just give us.
Okay, I prefer to say ligand.
We know that word's going to come up. So
it is.
Maybe we should
I don't like
start
ligand. I think it's an American way.
All right.
I guess being born in the UK, raised by an English family, I'm kind of like
accept the criticism.
ligand.
Oh, I forgot you're
No, it's
British.
okay. It's okay.
UK
But-
import.
Just like this
ligand is-
instrument here, so it's
Yeah.
not too bad. But yeah, basically, so in case you don't know what a ligand is, it's essentially a molecule that binds to something. In my case, I look at metals, in particular for my ligands that I've made, aluminium and zinc. So I've made...
So let me just break that down. Sorry.
Yes.
So you're saying you made a chemical.
Yes.
Yeah. And it binds to.
aluminium, I've got two that bind to aluminium and
Yep.
fluoresce, and another one that binds to zinc and fluorescence.
Yeah. So when these molecules that you've designed bind to these, attach to these
Yep.
metals, they fluoresce. Yeah.
Yep, they fluoresce.
Give off light.
Yeah, they essentially give off
Like a
light.
glow
where
stick.
Yeah, like literally is that sort of way how a glow stick works, if you like. So you essentially excite it with the light, either by leaving it out all day, and then when it comes to night or a reaction happens, then it actually essentially glows,
Yeah.
which is what happens with my ligands when they bind to, say, metals. So yeah, it's
Yeah.
pretty cool. And we can use that for applications for biology and medicine because we can essentially trace these metals in cells and for whatever experiments you biochemists want to do, as well as we can test these in river water samples as well so that we can detect potentially toxic metals from waterways so that we can kind of say, hey, let's avoid this waterway or clean it out so that all these animals and algae and environment essentially doesn't die on us, which is kind of the application towards it.
So let's just zoom in on, and this might get a little science heavy, so we'll try to unpack it as much as possible.
Yes.
On what makes a molecule fluorescent.
Yeah, and maybe like
Because a lot of
linked to that
people-
is how do you design a molecule that
Yeah,
fluoresces as
because like a lot of people, we talked about glow sticks, but they might not know at a sort of like atomic level
Oh, yeah,
Or-
well, this...
or
is it-
molecular level what happens to it. What are like some broad things that, like, or general things that are there that make a molecule fluorescent?
Yeah, well, it's essentially quantum physics, really. What essentially you get, unlike when you see color or colorimetric analysis, as we like to call it because we like to sound fancy. So with color, you're essentially getting a molecule or whatever you're doing, and it's essentially absorbing light, and whatever it doesn't absorb is being reflected back out, and that's the color that you see.
These gloves
They
reflect blue light
and absorb essentially every
everything
other.
else.
And
Yep.
that's colorimetric.
And that's colorimetric, so that's to do with color, hence in the
Yeah.
name. We just like to say colorimetric because we like to sound important.
Just like all the other fancy titles he attaches to himself. Right.
Exactly. Whereas fluorescence, you're actually exciting an electron into a higher energy state, and then it's dropping, and what it does there is actually release light from itself.
So let's just unpack that for the.
Yep.
You can tell me and him just got excited because we both teach in a unit called ACP and we have to kind of explain this to students. And we're like,
So an atom has a core, which is
Well, the nucleus, yeah.
Yep, the nucleus, and then it has
They're
electrons.
not spinning.
No, it has
Don't do that.
electrons orbiting.
Don't do that.
They're
not
like
orbiting.
electron
They're not
clouds.
orbiting. They're in an electron cloud. They're
Yeah,
not orbiting at
electron
all.
clouds.
Thank you.
No, but
Sorry, you almost gave me a heart
wait,
attack.
how dare you? Wait, but those clouds are just clouds of probability, right?
Yep,
They have
sure.
different shaped clouds for different orbits, whatever.
Different orbitals, yep.
Yeah.
So
And what happens is, maybe you can explain this, is when the light hits an electron.
A laser, isn't it, in this case?
Yeah, but it's
well,
photon,
it
dude.
doesn't necessarily have to be a laser. It just has to be
Just
a
light.
source of electromagnetic
Okay, yeah.
radiation. So it could even be UV.
Oh, okay.
So a lot of these are excited by UV, yeah.
Yeah, so let's just say light, right? A photon, which is the constituent of light, hits this electron, and that packet of light basically has energy, right? And so it excites the electron, and all of a sudden it goes to a higher orbital or a higher level of energy.
Higher energy state.
Energy
yeah.
state. And then when it comes back down, it gets rid of some of that, a different
It essentially gets
light.
rid of some of that energy and it gives off a different color light.
And how does it get rid of that energy? It lets off another photon.
Yes.
And that's what the light we're seeing is.
Yep, exactly, and that's what we're seeing, and it'll be at a different wavelength, a lower energy wavelength, so shorter wavelengths, higher energy. and longer wavelengths, lower energy, and
Can you
essentially.
link that to the colors of?
So that's fluorometric, right?
So that's fluorometric,
electric
yeah.
analysis,
Yeah.
when you excite it.
No, what I mean by that is like the shorter wavelengths are like
Yeah.
blue and
Yeah, well, the shorter wavelengths, yeah, they're blue, and then
the
longer
longer
ones,
wavelengths are like
red and whatnot, orange.
so,
Yep.
Yep.
So then, how do you? So that's what makes a molecule fluoresce.
So
Yes.
that's the electrons getting excited. So then is it like double bonds in a molecule?
To try and get it, so my design is essentially get a lot of pi conjugation, as it's
Oh,
called.
come on, bro. Break
Yeah,
it
yeah,
down.
I'll break it down. I wasn't just going to leave it there and be like, oh, they should know. The swine should know. So essentially that's when you go at a double bond, a single bond, a double bond, a single bond, a double bond, a single bond, just repeating. So if you're familiar with aromatic molecules, benzenes,
we're not.
they
There's
don't
a
necessarily have to
circle.
be.
There's
They're
a circle
circles
shape.
of what I essentially said, double bond, single bond, double bond, single bond. That essentially allows for electrons to resonate or migrate around the
You
molecule.
shared, yeah.
And then normally what I do is get something like an amino acid, which is found in biology, bound
in
to metals
proteins, yep.
naturally. So in proteins, for example, or enzymes, yeah. And so essentially that's my general design, at least so far.
So what you have is this ring, for instance, right,
Yes.
that has this double bond, single bond, double bond, single bond.
Yes.
And then attached to that, you have something that actually detects the metal, like an amino acid.
Well, that binds to the metal, yeah.
Yeah,
So yeah, I do
So
what's
it binds to the metal. So in
Yep.
this amino acid, you choose amino acids because they are found in proteins that typically use metals
Yep.
in their reactions.
and they're cheap.
And they're cheap. That's really cool. I didn't know that. So you're basically putting
And-
something that fluoresces and attaching it to something that can attach to.
Yep, that's nice and cheap, and also it's not going to kill a cell because if it says this is shift-based condensation reaction,
Oh,
it's a
what the-
don't worry about that. It's a condensation reaction, which means it gives off water. As a result, if you know anything about Le Chatelier's principles, you don't have to, don't stress.
We don't.
That's fine.
You're
That's
just
fine.
showing off now.
I don't mean to be.
This is what
but
wanky scientists do when they want to hide behind like their fancy terms. Bro. Break it down.
So essentially what it means is that the shift bases, which is the molecules that I make,
What
the
hell
general
is a
name.
shift base,
Don't,
bro?
don't worry about it. You don't need to know. They reverse when they're in aqueous solution, so water. So stop this. So they break down when they're in water. So that's not necessarily great for biology because biology is full of water. So I often try and use another reason for using amino acids for my sake is say that this bond breaks and we're left with our precursors, so our amino acids. And our fluorophore, they need to be innocuous to a cell line so they don't kill it as well.
So
Yep.
basically, tell me if I understood this because you confused the shit out of me too.
Sorry.
Right? So what you're saying is basically by using an amino acid and this like benzene ring or that fluoresces, if that gets broken down, then the products aren't necessarily toxic to the cells.
So it's still practical.
Yeah, that's important. Obviously, yeah, like you said, because that gives practicability to your
Exactly.
ligand.
Yep.
Yeah.
Ligand.
Don't say ligand.
Okay,
please.
so why detect? You mentioned waterways, but let's just focus a little bit on biological applications.
Here we go, coming from biologists.
Yeah.
Let's
Let's
you can tell that's what we're interested in, right?
Yeah.
Let's just make your research actually interesting. So let's talk about the biology.
Yeah, so why are you getting paid to produce this research?
He's not getting paid.
I'm on a scholarship.
Yeah,
but
he is.
Yeah.
Oh, he's
Yeah. Man, I did well enough in my first year to earn a
did
scholarship.
great.
So,
Yeah, good on you.
so essentially the applications in terms of, I guess, medical, more specifically to biological, would be, so I think the first one that kind of comes up is Alzheimer's disease. There's a lot of research into Alzheimer's disease about metals that potentially cause it. One of them that's being studied, well, a lot of the main ones have been studied for that are copper, or copper 1 specifically, zinc and iron. But there's also aluminium as well, and that's still in quite a bit of debate as to whether or not it actually does cause Alzheimer's disease. But nonetheless, I've made a fluorescent probe for aluminium so that those people, the biologists, the medical scientists who do want to use a probe so that they can track aluminium so that they can see whether or not it does cause Alzheimer's disease, etc., they can essentially use my probe and detect it inside of a cell and trace aluminium, potentially linking it to Alzheimer's.
So just talk a little bit about how metals might be involved in diseases like Alzheimer's. Because when you say,
actually, sorry, just on the manufacturing thing. So you just mentioned, I wanted to bring up something related to what you were just saying.
Yep.
So you just mentioned that if you're developing this technique to make this fluorescent probe that's quite cheap,
Yes.
then that is going to be quite easy for a lot of people to make themselves.
Yeah, that's another thing. And it's a high yield. It's like
Yeah.
97% yield, which is phenomenal.
Yeah.
So in terms of an industrial application, you know, I mean, why wouldn't you? Yeah, and it doesn't even require heat. I do it at room temperature for about 6 hours when I
Yeah.
do a synthesis of it. It's really
But the
easy with one
value in
step.
the value in that
Yep.
is that it's really like it democratizes science, right? Because now people can make this like ligand.
Yep.
They can produce it and do the type of studies on diseases that have significance.
Yep.
And do it cheaply. We had on Meow Ludo the other day, and some guys from Biofoundry, his lab that he runs is just a biohacking lab, and they've just developed a way to really cheaply make their own DNA ladders for the same reason. Like DNA ladders cost molecular biologists lots and lots of money. Yeah. So having a cheap ways of producing your own ladders is really beneficial.
Yep.
And I guess it's very similar with a ligand like this.
Yeah, with a ligand like this, yeah.
It
Yeah.
probably increases the amount of research that can get done on all the diseases that
Yep.
your
And it al-
ligand can detect.
Yeah, and it also means that I guess people who don't get too much funding can still afford to use a molecular probe such as mine, because it should, in theory, still remain nice and cheap if it does go on the market and get patented. But I think that might be somewhat daydreamy, but we'll see what happens.
Yeah,
That's-
but
that's-
still, that's
that's
a
the end goal though,
significant.
yeah.
That's a very like if you're publishing this data, right, and people
Aiming
are
to,
reading
yeah.
this and sitting, and they could make their own ligand, and that would be incredibly beneficial for the future of research.
Exactly. So my research essentially helps out a whole bunch of other ones. Is kind of the aim of it.
No, seriously, that's awesome.
Yeah.
Do you want to ask your question again?
Yeah,
that I
but-
so rudely interrupted you on.
Yeah, but do you want like a private room? Because you're like really...
No, that was good. I was vibing on that because I saw that thing at Biofoundry the other day, and I'm like, this is all, we're always whining about how small our budgets are.
Yeah.
So looking for these ways to like shortcut solutions, so you're
I
not paying
agree,
these
yeah.
big companies, is actually really beneficial.
Yeah, well, I said I don't see any point in doing research if it's not going to have some sort of application that's easy to use, I suppose, yeah.
No.
Yeah, that's...
Sorry, you were going to
I
ask
was-
about the significance of the diseases.
No, what do you ask him? Yeah, tell us about, like, not the significance, but tell us about, because you mentioned that these different metals like copper, iron,
Yep.
and potentially aluminium are involved in disease states like Alzheimer's. What sort of role would they play? I mean, what do metals do in biology? Besides like hemoglobin, which like traps oxygen in
enzymes,
red
yeah.
blood cells, yeah. But what about like in the brain?
So, just getting specific to Alzheimer's, because I know a lot more about that. So, copper, zinc, and iron are frequently found in the middle of these senile plaques formed by A-beta amyloid peptide. So, essentially what that is you get a whole bunch of peptides, so a sequence of amino acids. It's almost like a short protein. And due to these metals, or at least so far it looks like, due to these metals, they seem to stack on top of each other and keep on stacking, forming these big plaques, as they're called. And they essentially mess up your brain and get into your memory and whatnot, parts of your brain. And they can cause Alzheimer's disease, at least that's one of the lead ways. There's also the whole redox effects. This is probably a bit more technical. So metals like iron and copper. Copper exists as copper 1, copper 2. Essentially means it can lose and gain electrons. Same with iron, but with iron 2 and iron 3, it can lose and gain electrons, becoming iron 3, iron 2. And so this has essentially led to the oxidative stress, as it's called, that is believed to cause Alzheimer's as well. So this is essentially where, due to these redox potentials, as we call them, so reduction, oxidation, loss or gain of electron potentials, reactive oxygen species can be formed, which essentially can go on and react with biomolecules like proteins, lipids, and interfere with brain chemistry that way, and lead to the neurodegeneration or breakup of the brain matter. developing Alzheimer's disease. So there's another one, but there's heaps. Like I said, even if you're just studying enzymes, it's kind of good to get an idea about what metals are doing. There's even stuff like, I think, what Wilson's disease also involves metals. There's, well, even, see, like even action potentials, as you guys know, essentially how your nerves work. They work by influxes of potassium and sodium. Once again, they're both metals. And how do they find that out? Probably by something tracking it, potentially a fluorescent tag. I'm not too sure, but it's an easy application to use to do that. So, yeah. Because didn't even you track sodium or something, wasn't it, Hamid?
Yeah, well, that's my research.
Did you use a fluorescent tagger?
Yeah, I use a fluorescent tag.
There you go.
So does that mean that Hamid's an inorganic fluorochemist as well?
I just use it, dude. I don't attach fancy
You can call
titles.
yourself whatever you want, I find, and that's what I've done. So.